Today's episode of Science Spotlight is sponsored by fellow science geeks, the Irons team at Keller Williams Lake Nona.
This is the periodic table, noble gases stable, halogens and alkali react aggressively.
Good morning, Lake Nona, and welcome back to another episode of Science Spotlight. My name is Gabby Irons, and today we are going to be diving into quite possibly the best field of science there is, aka my favorite, microbiology. Microbiology is the study of microorganisms. Microorganisms are things that are too small to see with the naked eye, like bacteria, fungi, and viruses. Today, we are going to be swabbing Lake Nona High School to see just how many microorganisms we can find. The first thing we're going to do here is sterilize our glass vials, which we will use to store our swabs. We want to make sure everything is sterile to avoid contamination that prevents us from stating that the bacteria we see is really from the school and not from somewhere like my hands. We're going to fill these vials with 2 milliliters of sterile saline to keep the cells that we sample happy and healthy. After we've prepared our vials, the next thing we're going to do is collect our samples. For this test, we swabbed the school's doors, toilet seats, bathroom sinks, computer keyboards, trash cans, and stair railings. And now it's time for this week's quiz. The question is, which of the following microbes caused the Black Death? Is it A, E. coli, B, Ebola, C, Y. pestis, or D, S. pyogenes, or strep? The answer is C, Yersinia pestis. This bacteria is very famous for causing the plague that struck Europe beginning in 1347, killing up to 200 million people. After we've collected all our swabs, it's time to plate. Each plate, which is what we call a petri dish after it has been filled, contains agar, or nutrients which act as a type of food for the microbes. We're going to plate our samples on 3 different types of agar: eosin methylene blue, tryptic soy, and luria broth. In order to get our microbes on the plate, we are going to use a technique called streaking to isolate a pure strain from a single species of microorganism and get colonies. Now, after I've streaked and taped up all my plates, they're ready to incubate at 37 degrees Celsius or body temperature.
One eternity later.
About a week later, we've got growth. Let's check it out. The most growth, expectedly, was seen on the trash cans. And yes, this is what comes off the part of the trash can that you touch. We've also got significant growth on the toilets and bathroom sinks.
This is embarrassing.
The handles to hallway doors and inside of these doors also show some colonies. Notice how we got 2 swabs or trials for each location. This was to ensure that the microbes we see are not just there by chance. Overall, our results suggest that our school is kinda gross, but there probably isn't anything out there that'll harm you. And that's all we have for you today. Tune in next week for more Science Spotlight. If you'd like to see bloopers from today's episode or just the crew and I goofing around, visit our YouTube channel, and of course, stay nerdy, Lake Nona. P.S. Don't forget to wash your hands.